this is just a picture of the genome and its repeated sequences. And these abundant transcripts are called SIRS, which stands for herpes simplex uRNAs. And they're named that because, as you'll see in a minute, they look just like the uRNAs that are in our lysosomes. That's clearly where the blood is from. So there are 7 of these, and as I already told you, they're the most abundant viral transcripts. I'll show you this in a moment, that they look just like splicing SNRs. I told you that they cause, that the state then very often progresses to T-cell leukemias and lymphomas. What we work with in the lab are monocyte CD8 T cells, which once they're transformed by the virus, exhibit a mature activated phenotype. And very early on, the virologists working on this virus made deletions, trying to ask, what are these non-coding RNAs doing for the virus? And they didn't find any specific phenotype, like they couldn't go from latency to lytic or anything like that, at least in the lab. But what they did notice, and we notice this all the time, is that if you delete any of these SIRs, particularly this one and this one, which are the most abundant and most highly conserved, the cells in the lab grow more slowly. So somehow these SIRs are contributing to the proliferation rate of the RNAs. And here's one of these things about technology. So we've been working on this since 1986, when they were discovered. In the early 2000s, Heidi Cook, who was an MD PhD student in the lab, this was when microarrays came along and you could ask about gene expression, decided to ask, well, if I have cells transformed by the wild-type virus versus a virus that's lacking these 2 very abundant and very highly conserved SIRs, what will the host gene expression profile look like? And so she did that comparatively, and what she then ended up finding was that only a handful of genes were upregulated in the presence of SIRS, and all of those genes, and this just shows 4 of the 8, happen to be sort of hallmarks of T-cell signaling, genes that act early in the TCR signaling pathway, and here are some of them. So if you think about this, this makes sense for the virus. If the virus is going to go into a T-cell, and in a latent state, and want to exist there for months, if not years, before coming out to replicate, it's clearly to the advantage of the virus to keep the cells going, to keep them proliferating and activating the T cells is at least one way of doing this. But we had no idea of the molecular mechanism. Several years later, Damien Casala came along, and he was working on this problem. And what he realized, is what I'll show you in the next slide, was that some of these SIR sequences, I'm sorry, this sort of gets washed out here, were complementary to some host microRNAs. So first of all, again, I'm sorry that you can't really see what's here, but all of these, the reason that I say they look like splicing SNRs is they have trimethylcaps caps on their 5' ends, they have SN binding sites that bind to heterohaptomer proteins that all of the splicing snRNAs do. They have 3' terminal stem loops, etc. And what Damien realized was that for these 3 host microRNAs, the yellow are the seed sequences. Those are the sequences that we know are important for initiating a binding interaction, that these 2 could form interactions with the 5' end of these 2. Again, these are the most abundant and highly conserved. And the nucleotides that are in bold that you can see, you can't even see the other ones here, are the ones that are conserved. The ones that are absolutely conserved are in blue. And then he realized that this microRNA miR27 could interact up here. This is meant to show triple helix, but alternatives, double helix formation in miR16 down here. And what Damien went on to show after lots of investigations was that the functional consequence of this interaction was that the binding of miR27, the one that was over here in the last slide, to SIR1 inside the transformed T cells actually targets this particular microRNA for degradation. And you can see the half-life is slower and the levels then are lower. And there are a couple things about this that I want to comment on. First of all, this is a wonderful story in non-coding RNAs because you can think of it as non-coding RNA warfare, where the virus goes into the cell and makes a non-coding RNA that's going to feed into a non-coding RNA pathway in the host and do something, perturb it. The other thing that you should notice is that it's pretty clear that where the virus got these SIRS from was from the host splicing snRNAs. But notice they've done something completely different. Instead of using them as part of a splicing machine, they're using them as part of a degradation machine. So viruses do very, very clever things. But let me go on and summarize a little bit more about what Damien found. Okay, so conserved sequences complementary to cellular microRNAs. He could show that the RNP forms of the microRNP and the SNRP were associated in cell extracts. He did very elegant experiments where he changed the nucleotides in the base paired regions and showed that if you changed them, you didn't get interaction, but then you could make compensatory changes in the other strand and restore interactions, which is very good genetic evidence that there really is Watson-Crick base pairing. Turns out you can change the sequence of that particular region, get it to bind another microRNA, and then that microRNA is targeted for degradation. And finally, at that point, we didn't know too much about the targets of this microRNA miR-27, but two of them that were known are RONX1 and FOXO1. These are transcription factors implicated in T-cell malignancies. And Damien was able to show that they were upregulated. And about the same time that he published this work, there was a report from Amy Buck and Sebastian Pfeffer and colleagues that mouse CMV, cytomegalovirus, also downregulates this very same microRNA. So here's a beta herpes virus instead of a gamma herpes virus, and both of them are interested in this particular microRNA. Why is this? And I should say what this group has subsequently done is to show that it doesn't do it with a non-coding RNA. It uses the 3' end of what appears to be a messenger RNA. The microRNA interacts and is destabilized. And we don't know the details, just as we don't know that many details. But in any case, what Eric, the graduate student in the lab, wanted to do was to identify all the targets of miR-27 in the infected T cells and see if he could figure out why it is that this microRNA is of such interest to these viruses. And what he did was to use this beautiful technique that's been developed in Bob Darnell's lab across town at Rockefeller called HITS-CLIP for high throughput sequencing of cross-linked immunoprecipitated fragments. And the way this technique goes is that you take intact cells, you UV irradiate them. This forges covalent contacts between proteins and RNAs in the cell. So if this is a microRNP sitting on the 3' UTR of a messenger RNA, there will be cross-links between AGO and both the microRNA and the contacted messenger RNA. You then add a little bit of ribonuclease to chew away the ends of the messenger RNA. You immunoprecipitate with anti-AGO antibodies, and here we use two completely different types. You make a library by adding linkers and do high throughput sequencing, and then you filter the reads for those that have complementary to the seed sequence of the microRNA that you're interested in. And in this way, Eric came up with 59 different messenger RNAs that were targeted. Most of the target sites, 52% were in the 3' UTRs. And when one did Go analysis, gene ontology analysis on those messenger RNAs, it turns out that the most abundant class were in PCR receptor signaling pathway. So this was sort of what we expected, and we're very gratified to see this. What the other panels on the slide show is that miR-27 does indeed appear to be sort of a pleiotropic repressor, as I'll show you, of TCR-mediated T-cell activation. One thing that happens if you activate T-cells by TCR cross-linking is that you upregulate MAP kinases. Here's an example of P-junk. And you see that if cells are treated with miR-27, you get less upregulation than you do if they're treated with a scrambled microRNA. And likewise, CD69 induction, this is a cell surface marker of T-cell activation, is attenuated in the presence of miR-27. The next slide shows, on the left-hand side, the 10 top targets in terms of messenger RNAs of miR-27 in Eric's hits clip analysis. Half of these, the ones with stars, we don't know what their function is, but the other half are very interesting. This one and this one are transcription factors that have been implicated in humans in T-cell leukemias. And the three that are red, their functions are shown over here, and those are the ones that Eric studied and validated. And I'll just show you over here a bit about the functions. So SEMA7A is an extracellular signaling molecule that's put out on the surface and also secreted by T cells, and it interacts with integrin and also with plexin C1 on the surface of monocytes and causes them to increase their cytokine production. This molecule called GRAB2 is an intracellular signaling molecule. It goes to the nucleus of T cells and turns on genes implicated in T cell activation. It's also part of a negative feedback loop that keeps the cells from going into apoptosis, so it keeps them alive. And finally, you probably all know interferon gamma is secreted by activated T cells, and it recognizes receptors on macrophages and turns on the synthesis of components of the MHC, which then interact further with the PCR, et cetera, et cetera. So these are three very, very important molecules in T-cell signaling. And in every case, miR-27 represses them so that when the virus lowers the levels of miR-27, you expect these to go up. And I'll show you just a little bit of validation for one of these. Eric has done them for all three. The SEMA7A3' UTR, if you look at the hits clip peaks, there are lots of peaks. They correspond to a lot of different microRNAs binding to the 3' UTR. This is typical. There are two sites for miR-27, and Eric made reporters with luciferase and did mutagenesis and showed that you get the expected results. This is the more salient sort of slide here. So the amount of SEMA7A protein in human Jurkat cells can be decreased by adding this repressor, miR-27, by adding a locked nucleic acid, which binds up miR-27. You get the expected upregulation of SEMA7A. And the experiment over here is really important because it shows that this is all happening through this non-coding RNA called SIR1. So what Eric has done here is to knock down SIR1 in these cells with an antisense oligonucleotide. And so it's really missing. The levels of miR-16, which is called bind to SIR2, don't change, but the levels of miR-27 do in fact go up. And at the same time as it goes up, then the amount of the target, SEMA7A, in terms of protein production goes down. So that's the end of the data. And now I want to tell you about the surprise we got when Eric started looking at related viruses for what might be going on. So again, up here is a little bit of phylogeny. So this is the squirrel monkey I've been telling you about that gets infected by herpes virus Simuri and makes this non-coding RNA that lowers the levels of miR-27. Herpes virus Achilles is very closely related to Simuri. It infects spider monkeys instead, and it has non-coding RNAs. I'll show you where they are in just a moment, and does the same thing. These viruses over here, the Maka viruses, do not appear to encode any non-coding RNAs. but they're related. They also infect T cells. And they also, I should have emphasized over here, that both of these species, when they infect their natural host, just have an infection that makes lots of virus, but it doesn't cause any harm. It's only when it goes into related species that it causes these T-cell leukemias and lymphomas. Same is true of these viruses. So this one, the natural host is wildebeest, no problem, it just replicates, and sheep, no problem, it replicates. But when these viruses get into other related species, like goats and cows and pigs, they cause a disease that in Africa is called malignant catarrhal fever, which is a lethal T lymphoproliferative disease. And it's really quite an agricultural problem in Africa. If we now look at what's going on in the genomes of these viruses, I already told you that herpes virus cinerea has these 7 non-coding RNAs. These are the 2 highly conserved ones. The squirrel monkey only has 2 of those, and there they are. And this one also binds miR-27. In the same region of the genome of these other viruses, they don't have non-coding RNAs, but what they seem to have done is to pick up from the host homologs of the protein targets of miR-27. So the red arrows are C7A homologs. The green arrows are a transcription factor that acts on the to make interferon gamma, and here's a viral IL-10. So over the course of evolution, it seems like these viruses have solved the same problem. They want to upregulate the levels of these key T-cell signaling molecules inside cells, and they can do that either by lowering a microRNA that's a repressor, or by just picking up the protein coding gene for the targeted messenger RNAs. So that was a very surprising result that we certainly did not expect. And I'll just summarize here and then I'm done. So miR-27, we identified these targets and showed that most of the most enriched genes were in the T-cell activation and T-cell receptor signaling pathway. We validated some of these. I should mention earlier I said that there were other T-cell proteins that were upregulated. None of those seem to be direct targets in miR-27. We think they're indirect targets. But in any case, these viruses target multiple components of a conserved pathway in order to make sure that their latently infected cells survive and are there when the virus wants to be induced. But the remarkable finding is that during viral evolution, you can solve this problem in 2 completely different ways, either by picking up a non-coding RNA from your host and changing it to do what you want it to do, or just picking up the protein coding genes and have them make more. And one conundrum in the literature that was solved, I told you before that mouse CMV lowers the amount of miR-27. It was very surprising when people looked at human CMV a couple of years ago and found that it doesn't do that. But now looking back at human CMV, we see that it instead encodes a viral IL-10 homolog, which is a target of miR-27. So it appears that this strategy of either picking up a non-coding RNA or picking up a protein coding gene may have been repeated in evolution and may be something that we'll see more of as we look at even more viruses. So in the end, I just want to say thanks to, this is Jess Brown, who solved the crystal structure of MALAT1-EME, a lot of help from Max Alonson, a Yale undergraduate, graduated last year. This is Kevin Kowski, who's done all of our bioinformatic work on identifying triple VOCs, and Eric Ruhl, whose work I just told you about last, and thanks to our other funding sources, and particularly to the real virologists at Yale that helped educate us about what's going on with these viruses. So thank you very much.